Hello my dear students, I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. So, we are learning about this subject called Pharmaceutical Biotechnology, and we have almost already covered 3 units of Pharmaceutical Biotechnology. This is the 4th unit. From today we are going to start. This unit is very important, and after completion of this unit, you are going to get so much information, so much knowledge that you will be able to utilize in detection of various diseases. My dear students, I hope all of you are aware there are huge amounts of viral or bacterial or microbial diseases are actually present in our world. Still the date we do not have sufficient number of detection kit to that is detect those diseases if somebody got infected. So, this unit will help you a lot to understand how you can make your own that is a diagnostic kit to detect various viral or other kinds of diseases. Today, I am going to tell you about the ELISA. The name is so much popular to you. I hope when you are actually learning about HIV, you got to know this word ELISA. I am going to tell you everything about ELISA on today. So, today I am going to tell you some basic terms. Then what is ELISA, principle of ELISA, how it is actually working, then what are the equipments of ELISA, what are the types of ELISA, advantages, disadvantages, and what are the limitations. So, let me start with some basic terms. My dear students, in unit 3 we have seen antigen and we also have seen antibody. Antigen actually is the foreign substance that is not a part of our body, it is coming from the outside of our body. Sometimes our body is also creating some antigen. For that, in our body we have antibody. Here we are actually going to do the engineering of those molecules. Now, antigen, a molecule that elicits the production of antibodies when introduced into the body. Obviously, when some antigen will enter, your body will produce. And what is antibodies? A protein produced in response to the antigenic stimuli. This specific structure of antigen, it is actually binding with the antibody. All of you are aware antibody will be Y shaped structure. Here I have given antibody, this is the picture of antibody. Okay. Antigen will be, antigen structure will be might be any kind of structure might be present, but these things will be in Y shape. Okay. So, and what is enzyme conjugate? An enzyme that is attached irreversibly to an antibody, that is horseradish peroxide HRPO. I am going to tell you more about these things. Let me tell you something basic terms, some other things like chromogen. It is a chemical alters color as a result of enzymatic reaction with substrate. Coloration is used to the signal. If a reaction has happened, if a reaction is completed with especially presence of enzyme, in that case it will give some color. There is something called stopping. This is a process of the process of stopping action of an enzyme of a substrate. What is reading, sir? Spectrophotometric measurement of a color developed in ELISA. Okay. For example, here you can see some reaction is happening with the enzyme and your other substances, some substrate actually get involved, and ultimately you got some product. So, chromogen will give the ultimate signal that yes, a color, it will give the color that yes, it was actually happened, anti-reaction actually happened. Such things you can get. Another thing is very important to understand our ELISA and our upcoming also lectures like blotting techniques also. Another very important function is that is adsorption. It is a process of adding antigen and antibody diluted in buffer. So, it attaches to the solid phase of the on incubation. Okay, solid phase, here you can see this is the surface. Okay. So, that is in this process we actually adding antigen antibody diluted into the buffer. Okay. This is adsorption. Another thing actually called absorption. Sometimes we actually confuse between adsorption and absorption. In absorption, whatever the molecules or whatever the antigen antibody we actually give, in absorption it will get diffused, and in adsorption it will be in the surface. Okay. So, I hope it is very much clear to you all. What is washing, sir? It is just simple flooding or emptying walls with buffer solution to separate the bound from unbound reagent in the ELISA. So, when we do ELISA test, For example, these are few antigen or these are vice versa, these are few antibody. So, if it is a antigen, so what will happen? If it is an antigen, what will happen? Some antibody will come here and it will bind, it will bind. So, after that we have to do some washing, because if any other extra chemical may come here, it will create some that is a problem in our reaction. So, we are doing actually washing. Okay. So, I hope it is very much clear to all about the basic terms whatever I have told you in these 2 slides. Now, let me take to you to the main part. What is ELISA? So, its full form is enzyme linked immunosorbent assay. It is a plate based assay technique, plate like this, plate based assay technique designed for detecting and quantifying the concentration of analyte. Analyte might be anything, it might be your antigen, it might be antibody, it might be protein, or it might be glycoprotein. Any of the 4 things might be present. For example, virus. In case of virus, obviously the antigen will be there, but when in your body after the introduction of the virus, some antibody will be created. I want to know how much antibody developed in the patient's body, so that I can analyze his immune response. In that case, we have to take the antibody from the patient's blood, or in the other case somebody got infected recently, in his blood virus is present, and in that scenario we have to take the virus particularly as an antigen. Okay. Sometimes we can also detect some protein. Okay. What are the examples? Some examples are like in HIV infection we are actually using, pregnancy test we are actually using, measurement of cytokines we are actually using these things. Here is a very good picture of a marketed that is a diagnostic kit. Its name is AKtU ELISA HIV 1 and 2 AV AG. So, this is actually one of a kit, it is that is actually used for the HIV detection. Okay, it is coming under the immunodiagnostic. This is the company Diagnostic Automation Inc. They are actually that is developed or marketed this ELISA kit. Another you can see, this is the pregnancy detection kit. I hope many of you have seen this various ads on the television. And here is another example of kit, cytokine ELISA kit. See, in my previous lectures I have told you about the interferon, cytokines, how cytokines are playing a big role when a virus enter in our body and infect our cell. Our cell is actually informing other cells that yes, some virus has entered, you may get alert, you may ready, I got infected, you should be ready to fight against this virus. So, basically cytokines are actually playing a big role in signaling, telling the information, but cytokine are sometimes it will create some problem in our body like fever or various kinds of cytokine storms are actually happening. Due to that we are getting high temperature in our body. Okay. So, if in autoimmune disease or if a cytokine actually created in our body by mistake, in that case also we have to detect the presence of cytokine. So, for that you can see we have a lots of cytokine ELISA kits are also available. This is in Vitrogen, one of the pharma company, they have that is marketed this product. Now, what are the principle of ELISA? The principle is very simple, it is based on lock and key concept. I hope all of you have studied in your school days, also in B Pharm first year and second year about the lock and key theory. So, the antigen whatever is present, we call it as a key, or antibody what is actually happened, we call it as a lock. So, key benefits into the lock, enzyme conjugate substrate actually developed and bound to secondary antibody that bind the antibody antigen complex. So, whatever the antigen antibody complex will be actually created, from that we can analyze, from that we actually able to confirm that yes, this antigen actually present, due to that this complex is actually happening. For example, let me also tell you in a simple manner, you are trying to do some test, you actually given the antibody and this is the sample. So, if antibody and antigen both are present, both of them will form a complex. If it is not present, only antibody will be there. Okay. If it is present, then you will be getting one kinds of picture, one kinds of color or something, and if both the things are present, you will be getting different kinds of signal or different kinds of color. Okay. So, based on that you can able to confirm. Although many variants of ELISA have been developed and used different situation, there depends on the some basic elements. What are the basic elements? Coating or capture, direct or indirect immobilization of antigens to the surface of the polystrand microplates wells, or plate blocking, addition of irrelevant protein or other molecule cover all the unsaturated surface binding sites in the microplate tube, or probing, detection, incubation with the antigen specific antibody, or otherwise in the signal measurement, detection of the signal generated by the direct or secondary tag in the specific antibody. These are the various that is we can say elements, basic elements which are actually playing a big good role in ELISA. Now, what are the equipments? First is the microwell plate. You remember at the beginning of my lecture I have told you that is a plate based technique. So, flat bottom polystyrene plate, it will containing 8 by 12, 8 by 12 holding 350 microliter each, whatever there will be one hole, it will be like this. Okay, it will be like this, and it will be having this 350 microliter each. Another thing is called actually micro pipette. I hope you have used pipette in your chemistry activities. Here we are actually using micro pipette. Here you can see this is a very micro amount it will be giving to you this plate, micro pipette. And then washing device, manually operated washing devices are present, or it may be used partially where there is a risk that samples tested in ELISA contain infectious material. So, must be collected and subsequent disinfection. Okay. Now, come to very interesting point. What are the types of ELISA? What are the types of ELISA and how you can perform various types of ELISA? What are their advantages, disadvantages? Everything I am going to tell you. First of all, direct ELISA. Here you can see here this is the antigen. This is the antigen. Antigen is in the surface, and then you can see the primary antibody conjugate actually coming, some substrate actually created. In indirect ELISA, what is happening here? Antigen actually present here, some antibody is actually coming, another secondary antibody conjugate actually coming, another you can see two antibodies are present here, then some substrate actually created. Sandwich ELISA, here actually in both these cases we are observing that antigen actually in the surface, here the antibody will be in the surface, here will be the antigen, then again enter these two things, that is two antibodies will be there, and both the things will be do some reaction, and then you will be getting some in that is a signal as a substrate. In competitive ELISA, this is very interesting. In this case antigen will be happen. So, there will be something called inhibitor antigen and antibody, ultimately you will be getting some result. So, let me tell you more, it might be slightly confusing for all of you how they are actually working, how they are different. Let me tell you more in detail. So, direct ELISA, whatever I have told you here, these things direct ELISA. So, here the antigen actually immobilized directly onto the surface of a multi-wall microliter plate such as 96 polystyrene plate, and then complexed with the enzyme labeled primary antibody specific for the antigen. So, antigen actually immobilized this it is immobilized, it is present in the surface the antigen. Once the enzyme labeled primary antibody actually bind to the antigen, the conjugated primary antibody catalyzed a reaction with its respective substrate, resulting into a visible colorimetric output, these things by spectrophotometer or absorbance in micro plate reader. Direct ELISA is suitable for qualitative and also quantitative antigen detection in the sample of interest, also in antibody screening and also epitope mapping. Now, let me tell you more how it is actually happening. So, you can see this is the antigen, basically these are the sample, these are the sample, Okay. These are the antigen, it is present in this your plate, microliter plate in the surface. What is actually happening? You are actually giving the antibody. For example, HRP labeled primary antibody you are actually given, and then it is actually bounded, and then after that, after they are the mixing, there will be some color product will be created. For example, you can see this substance TMB substrate actually given to see the color, to see the what kind of things actually happening. You can see from this it has given this color, or otherwise you can also take some sample from that, or you can analyze by using spectrophotometric method, and through that you will be able to confirm that yes, this antigen actually present. If this antigen is not present, what will happen? No reaction, nothing will be happen. Ultimately, same color will be remain. Okay. This is called direct ELISA. Advantages: quick, because only one antibody and fewer steps are actually involved. Cross reactivity of secondary antibodies that is eliminated, but yes, there are some disadvantages. What are the disadvantages? Immune activity of primary antibody might be adversely be affected by labeling with the reporter enzymes or tags. Labeling primary antibody for each specific ELISA system is time consuming and expensive, and yes, limited number of the conjugated primary antibody will be available commercially. So, these are the few disadvantages. Now, we have learned about the direct ELISA. Now, in my previous slide here I have also told you about the indirect ELISA. So, let me tell you about indirect ELISA. Yeah, in this area antigen or sample actually added to the plate. You can see this is your microplate. Here your sample or antigen actually given to the plate. Then blocking buffer actually added to the block, remaining the protein binding sites. You can see buffer was added. Buffer was actually added in that area. Then next, a suitable primary antibody is actually added. So, obviously, it is antigen, there will be some antibody for that, a suitable antibody you have actually added. Then what you are doing? You are actually adding a secondary antibody, HPRO conjugate is then added to the in which recognize and binds to the primary antibody. Okay. This this is the secondary antibody. This is the secondary antibody. So, why actually you have added? To get the result much more clear. Okay. So, entire the HPRO secondary antibody is a complex, it is a complex, both the things are actually added. So, you may imagine antigen is present, antibody has binded with this one. After that, the secondary antibody it is also binding with this one. Now, if antigen is not present, what will happen? No binding will be happen, it will be not bind with this, and ultimately this will be also not happen. And ultimately, some TMB substrate will be added to convert the HPRO HRPU to detectable form. How? See, this will be the colored product. So, what is the red color you are observing? So, TMB substrate it will change its color. Color changes is the ultimate confirmation that yes, the entire experiment was completed successfully, and yes, this antigen is actually present. So, this is called indirect ELISA. Okay. What are the advantages? A wide variety of labeled secondary antibodies are that is available commercially. Versatile, because many primary antibodies can be made in one species and then same label secondary antibody can be used. Maximum immunogenicity of the primary antibody is retained because of not label, but yes, there are some disadvantages. Cross reactivity might occur with secondary antibody resulting non-specific signal, and yes, an extra incubation step is actually required for this procedure. These are the few disadvantages. So, this is something about the indirect ELISA. Okay. Now, I have told you another two kinds of ELISA, sandwich ELISA. Now, let me tell you about the sandwich ELISA. Let me go to the slide. Yeah. So, you have to understand that in this type of ELISA, two antibodies, a capture antibody and a detective antibodies are actually used. Two antibodies are actually using. Here also in indirect ELISA, we actually use two different antibody, but here the picture is slightly different. Here capture antibody and detection antibody actually using. The purpose of any ELISA is to detect the presence of the target antigen in a sample. In sandwich ELISA, the target antigen actually bound between capture antibody and detection antibody. I will tell you with the picture. So, whatever the antigen, it will be in between the capture antibody and detection antibody. The capture antibody is immobilized on surface, while the detection antibody, that is a conjugate of enzyme or your fluorophore level, is applied as a last step before quantification. The two antibodies used in sandwich ELISA must be paired and tested before use. How? See, this is the picture. This is the picture. So, this is the capture antibody. This is the capture antibody. Then you have some virus or something you are trying to identify. Then you have added that antigen. You have added that antigen. After that, biotin level detection antibody is actually you have added. Now, that antibody again binding with this, binding with your particular antigen. Now, what next? The next is streptavidin HRPO. You have seen in previous my that is indirect ELISA also how HPRO playing a big role, it is changing its color. Similarly, here also you can see that is a streptavidin HPRO, when these things are actually added, and after that you have added TMB substrate. This TMB substrate will bind with this and it will give you some color. This color conformation, the color changes the from this to this is the confirmation that yes, this antigen actually present. If antigen is not present, in that scenario such kind of reaction will be not happen, and then this color will be remain. Ultimately, we got this color, it means the antigen actually present. This is sandwich ELISA. What are the advantages? Highly sensitive and highly specific for target antigen, as two antibodies are actually used to capture and detection, and then different detection methods can be used with the same capture antibody. What are the disadvantages? It requires more optimization to identify antibody pair and ensure that there is a limited cross reactivity between the capture and detection antibody. Now, another thing, I hope you are very much clear about direct, indirect, sandwich. Now, come to the last types of ELISA called competitive ELISA. Let me tell you more about the competitive ELISA. So, it is a technique used for estimation of the antibodies present in a specimen such as serum. What is the principle? The principle of test is that two specific antibody, one conjugate with the enzyme and the other one present in the test in the serum. If serum is positive, for antibody are used. So, competition actually occurred between the two antibodies for the same antigen. There are two antibodies for the same antigen, a competition will be happen. So, appearance of color change indicate the negative test, absence of antibodies, while the absence of the color indicates the positive test, presence of antibodies. Okay. So, in a sample or in a serum, if certain antibody is actually present, okay, all of you are aware what is serum, plasma, serum, blood, what are the difference? I have all of you are aware about these things. So, you have blood, you have taken the plasma, then you have separated the serum from that, and then you have tried to add two specific that is a substance with that. So, what will happen? A competition will occur between the two antibodies for the same antigen. So, the central event of the competitive ELISA is that competitive binding process executed by the original antigen or sample antigen. Okay. So, you can see this is the step 1 where you have added the sample. In step 2 you have added two different antibodies, and after that you have done some washing, and later on you have done the got the substrate. So, this is competitive ELISA. What are the advantages? High specificity, since two antibodies are used and the antigen analyte is specifically captured and detected. Suitability for complex sample, and it is highly flexible that is and sensitivity, since both direct and indirect detection methods can be used. And but yes, there are some disadvantages. What are the disadvantages? ELISA technique can be adapted to competitive format. The same limitation described for the ELISA technique. Okay. So, these are some disadvantages. So, this is something about the ELISA. Now, if we talk the entire that is advantages of ELISA. So, reagents are really cheap and that is it has a very long that is shelf life. It is highly specific and sensitive, no radiation or hazard actually occur, and easy to perform. Equipments are widely available. It can be used in a variety of infection, and it can be used most of the type of biological product like plasma, serum, urine or cell extract. Okay. So, that is why it is highly popular, all the 4 different kinds of ELISA whatever I have told you. But yes, disadvantages, what are the disadvantages? Measure enzyme activity can be more complex than measurement of the activity. Enzyme activity may be affected by plasma constitute, kits might be not cheap and very specific particular antigen, but would not recognize the other antigen, and false positive, false negative is also possible in case of mutated or altered antigen. Okay, false positive results also happen. You might have listened in recently rapid antigen test or various kinds of antibody test for SARS-CoV-2. Patients are getting false positive results. Why? Because of this problem. Limitations, results are not absolute in maximum times sometimes. Antigens must be available in poor product interference, and yes, concentration may be unclear. False positive, whatever I have told. So, these are some limitations of our this ELISA test. So, today we have seen the basic terms. After understanding the basic terms, we have seen how these basic terms are used in ELISA. Then I have told the principal, equipments, types, advantages, disadvantages and limitation of ELISA. So, that is it for my side for today. In next lecture, I am going to tell you something more interesting thing, some more advanced technique by using which technique you can check, you can test, you can diagnose some presence of some protein or some DNA or RNA in a certain mixture. I am going to tell you again some basic terms, then I will tell you how these basic terms are involved in human blotting technique. Then I will tell you southern blotting, northern blotting and western blotting. So, these are the few interesting things we are going to learn in next lecture. So, that is it from my side for today. I hope all of you have enjoyed this lecture. So, thank you for joining. We may meet in our next lecture. Till then, God bless you all the best.